Hi! In this presentation, we'll be discussing viruses. Viruses are still a bit of a mystery to us today because they can be extremely dangerous and nearly impossible to get rid of. So what we're going to talk about in this presentation is the characteristics of viruses. So before we talk about the characteristics of viruses, we're going to talk about a little bit of history on how viruses were discovered. So this is the story of viruses. In the 1890s, a Russian biologist by the name of Dmitry Ivanovsky was noticing that farmers were losing crops to a strange disease. The disease turned the leaves of tobacco plants from green to yellow, and it sort of made this mosaic pattern that you can see on the leaf in this picture. Eventually, the leaf would turn completely yellow and the plant would die. So what Dmitry did was he extracted the juice from the leaves, so he sort of crushed the leaves up until they formed this liquid juice. And then he took that juice and he added it to the leaves of healthy plants. What he found was that after being exposed to the juice, the healthy plants would then develop the disease. And so he was able to determine that whatever disease this was, it was contagious. So he was trying to figure out what exactly is in this juice that can be spread from one plant to the other? What is it in one plant that is making it sick? And so he filtered the tiniest cells out of the juice. And so we went through this filtration process where the actual cells of the plant that had been crushed up and turned into liquid were filtered out. Still, when he added that juice to a healthy plant, it would still get infected, it would still die. So what he determined was that you cannot see whatever particles were in there that were making the plants sick, but he knew that they were there. With help from other scientists, they were able to discover that there are these tiny particles that can cause disease. The word virus actually comes from the Latin word for poison, so that is what they coined this type of particle from. So what exactly is a virus? Well, viruses are particles made up of nucleic acids, proteins, and sometimes lipids. It's really important to remember that they are considered non-living. So rather than calling them cells, you should call them particles, because you don't want to imply that they are a living organism, because right now we do not consider them living. Although they contain some of the same organic molecules as living things, they aren't considered living. They cannot reproduce without the help of a host. We're going to talk a little bit more about how viruses do reproduce and how they use their hosts. They also cannot perform metabolic processes. Now, a metabolic process is what organisms do to get their energy. So viruses cannot do photosynthesis or respiration because they don't have the right equipment to do so. They can't express their DNA themselves because they do not have ribosomes, so they're unable to make protein. And proteins are the way in which DNA is expressed. So again, in order to express the information in their DNA, they need to have a host. So they use a host to reproduce, they use the host's energy, and it relies on its host for expressing its genetic information. So here is a picture of a couple of virus structures. You can see that viruses can kind of come in a lot of different shapes and sizes. They have, all of them, even though they're different, they all have some things in common, like a protein coat and genetic information. So what I would like you to do at this point is pause the video and take down these few bullet points as notes. So, as you can see, viruses all contain what's called a capsid. Capsid is just a fancy word for a protein coat. So in this T4 bacteriophage, you see everything that's in green, that is protein, and it's sort of coating the virus. Again, here, this is the tobacco mosaic virus. This is the virus we were talking about in the first slide. Again, it has this protein coat. And then here's a flu virus. It's a slightly different shape. It's rounded, but again, it has this capsid. It has this protein coat. Proteins are kind of like, they can be considered similar to the Trojan horse, and the cell that the virus is attacking can be compared to the city of Troy. So here's how these proteins work, just like the Trojan horse did. When the virus latches onto the cell, the proteins sort of trick the cell into thinking that the virus is supposed to be there. So the proteins are compatible to the proteins on the cell that it is attacking. That way, the virus is able to latch on without the cell reacting to it and killing it. Then it's able to inject its DNA, just like the soldiers were able to crawl out of the Trojan horse and attack the cell. For this reason, viruses are really, really specific to their host, because the only way that they're able to latch on is if they have the right proteins on the outside of them that allow them to latch on. This is also why it's really common for some organisms to be carriers of viruses. So for example, a lot of birds carry some viruses that don't attack the bird because the bird cells are not compatible with the virus's protein. Instead, what happens is when we come into contact with those birds, the virus comes into our body, then the virus has the right kind of host that matches its protein coat. So something like the avian flu, that sort of virus has a protein coat that's compatible with human cells, but not with bird cells. So it doesn't kill the bird just sort of carries it around. It doesn't really become active until it comes into contact with human cells, and in that case it's able to attack. You can also see all of these cells have genetic information, and the genetic information can come in a couple different forms depending on the virus. It can be DNA, which we see in our first virus here, or it can be RNA, which we're seeing in the flu virus here, and in the tobacco virus too. Sometimes the DNA is single-stranded, and sometimes it's double-stranded. We'll talk more about what those terms mean when we talk about genetics, but for now, just know that the genetic information can come in a few different ways. So, now that we know a little bit more about the structure of the viruses, we're going to talk about how these different structures contribute to how the virus is able to affect. The viral infection can be one of two types of ways of infecting. There's something called the lytic cycle. Lytic infection is very fast, the viruses replicate really quickly, and then it kills the host cells right away. Another type of infection is a lysogenic infection, and in that situation, the virus replicates slowly, so it sort of stays dormant for some time. What I'm going to do now is I'm going to give you two slides that contain information on lytic cycles and lysogenic cycles. So what you should do is pause the video now and take a minute to write all of these bullets down. Now, pause the video again and write down the notes for the lysogenic infection. Now that you have the notes for the lytic and lysogenic cycles, I'm going to talk through those cycles with you as they appear in this visual. So first, let's talk about the lytic cycle. Now, in both cases, the way it starts off is the virus, let me just slide up here, the virus latches onto the cell, okay, so you can see that happening here. And then it inserts its DNA. You can see the small loop of DNA. The DNA forms a circle. Now, a couple things you should be aware of. This word bacteriophage, what that means is a bacteriophage is a virus that infects bacteria. So here we have a bacterial cell. However, in eukaryotic cells, like in humans, animals, and plants, other organisms like that, viruses still can attack in either a lytic or lysogenic way. Just for this diagram's sake, and the simplicity of the prokaryotic cell, we're going to look at how it infects a bacteria. For the lytic cycle, what happens is the virus takes over the bacterial cell. What it's doing is it's using the bacteria's equipment to replicate its own DNA. So instead of the bacteria replicating its DNA, the virus's DNA is being replicated instead. So what you get is a bunch of viruses being produced, more of these bacteriophages being produced inside the bacterial cell. using the bacteria's equipment. Then, once they're completely developed, they burst out of the cell like this, and the cell lyses, or bursts, explode. That's where the word lytic comes from. To lyse means to pretty much explode. So that's the lytic cycle. It's pretty destructive. It happens really fast. You see a lytic infection in the flu virus. Like when you catch the flu from somebody, it's not like you stay healthy for a few months and then all of a sudden you get the flu. It hits you pretty quickly because the flu virus follows a lytic cycle. The other way a virus can infect is called the lysogenic cycle. So it starts off the same way with the virus inserting its DNA. The DNA sort of forms this loop shape, but when it follows the lysogenic cycle, what it does is it slowly becomes integrated into the DNA. Now this word prophage, that's just a fancy word for the viral DNA. So in pink we have the virus's DNA, and then the blue is the bacteria's DNA. You can see it's just sort of become a part of it. So what happens is as the bacteria goes through its regular cycle, or in our case, in human cells, our cells are just going through their normal life cycles. Sometimes they are reproducing when needed. The viral DNA is just sort of hanging out there, inactive, just sort of becoming part of the DNA. But you can see that it's being replicated using the bacteria's tools. Eventually what happens is that at a certain period of time, whether it's due to an environmental factor or something else, a virus that infects in the lysogenic way enters into a lytic cycle. So after it has slowly been dormant in the cell's DNA for a really long time, something causes it to activate, and then it enters the lytic cycle, where it all of a sudden starts to replicate really fast, more viruses are made, and then the cell lyses. So that's the sort of infection that you see with HIV. You might see it with the herpes virus. A lot of sexually transmitted diseases follow lysogenic infections. And the reason they're able to spread so quickly is because they remain dormant in the body for a long period of time until some sort of environmental factor causes them to become active. For example, the herpes virus, you might have the herpes virus and not even know it until maybe you're under a lot of stress or something like that, and suddenly you get a cold sore on your lip. That's the herpes virus becoming active and killing the cells in your lip and spreading. So in both cycles, the cells end up dying. You end up having some sort of infection. Just in the lytic cycle, it happens really quickly, and in the lysogenic cycle, it happens over a long period of time. So, for your left side activity, what I would like you to do is create a Venn diagram comparing and contrasting bacteria and viruses. So in each side, you should have 4 points that are differences between bacteria and viruses. And then in the middle, make sure you leave yourself enough room to write 3 similarities between bacteria and viruses.